My name is Lillian Ko. I'm a program director in the Division of Cancer Biology at the National Cancer Institute, or NCI, and I'm delighted to welcome you all to our Cancer Moonshot seminar. I'd like to make a few points about today's seminar. In supporting the next generation of cancer researchers, today's cancer, In supporting the next generation of cancer researchers, today's Cancer Moonshot seminar will feature research from very talented early career scientists. And these are abstract-driven talks nominated by Cancer Moonshot investigators and selected by NCI staff. Before we get started, I just had a few bookkeeping items to review. Questions will be responded to at the end of the second presentation. So please enter your questions in the Zoom Q&A box, not the chat box. The audience, you can then prioritize the questions by clicking on the thumbs up button. There is closed captioning available. The closed captioning for today's webinar can be accessed by clicking on the live transcript option in the Zoom menu at the bottom of your screen. And instructions are also provided in the chat box. And a recording of today's webinar will be available a few weeks after today's webinar. And so again, please put your questions in the Q&A box, not in the chat box. Only if you need technical assistance, please use the chat box. And so now it is my great pleasure to introduce our featured moderator, Dr. Suzanne George from the Dana-Farber Cancer Institute. Dr. George is a medical oncologist and clinical investigator. She is an associate professor of medicine at the Harvard Medical School. And she is the clinical director of the Center for Sarcoma and Bone Oncology at Dana-Farber, which is one of the largest multidisciplinary sarcoma programs. Her clinical research expertise is in rare tumors, specifically sarcomas. She's internationally recognized as a clinical trialist in soft tissue sarcomas with specific interests in leiomyosarcoma and gastrointestinal stroma tumors. She has served as vice chair of the NCI Oncology Cooperative Group, or the Alliance for Clinical Trials in Oncology. And as vice chair of the Alliance, she has worked to develop cross-disease efforts for various young adults. And so with that, I'll hand it off to Dr. George. Thank you.
Great, Lillian, thank you so much. And thanks everyone for having me today. My colleague, Dr. Jenny Mack, was due to be the moderator, but at last minute was unable to come. So I appreciate the opportunity. Actually, I'm really excited to hear from our speakers, and I'm glad to be here today. So I'm going to introduce our first speaker, Dr. Rachel Blankson Isaka, and I'm going to apologize in advance to both of our speakers who I would ask to actually introduce their own proper pronunciation of their names after I give some background, if that's okay. So our first speaker is Dr. Isaka, who is a gastroenterologist and associate professor in the Catherine Smith Endowed Chair in Health Equity Research at the Fred Hutchinson Cancer Center and the University of Washington, where she provides care for patients with general GI disorders and GI complications of cancer and cancer treatment. She also is the director of the Fred Hutch UW Medicine Population Health Colorectal Cancer Screening Program. And her research efforts focus on early detection and prevention of colorectal cancer through increased screening, particularly among racial and ethnic minorities and individuals with lower socioeconomic status. But we're really looking forward to hearing her talk today, which is really focused on trying to overcome challenges associated with lack of transportation and access to screening colonoscopy. She has done some evaluation through an academic and industry partnership, executing a novel rideshare, non-emergency medical transportation intervention for patients with barriers to colonoscopy completion because of transportation. So I welcome you here today and really look forward to hearing about your work.
Thank you so much, Dr. George. And it's Isaka. It's a pleasure to join you all. I'm going to go ahead and share my slides with the audience so we can get started. Can everyone see that okay?
Yes, go ahead.
Okay, excellent. So it's my pleasure today to share our work, and this is titled "The Health System Provided Rideshare is Safe and Addresses Barriers to Colonoscopy Completion." So there is a significant and growing need for colonoscopy. Based on the most recent data, approximately 13.8 million colonoscopies are completed annually in the United States alone. And more than 75% of these are completed for colorectal cancer screening and surveillance. I think most in the audience are aware that colorectal cancer is the second leading cause of cancer deaths in the United States, with an estimated over 53,000 individuals this year alone who will ultimately die from this disease. And colorectal cancer is in large part preventable through screening, with colonoscopy being one modality by which we can do that. And certainly colonoscopy is needed for those who use non-invasive strategies as well. But unfortunately, 1 in 3 adults in the United States is not currently up to date with colon cancer screening. And in fact, it's estimated that 63% of colorectal cancer deaths are ultimately due to missed screenings. So I mentioned briefly some non-invasive strategies that do exist for colorectal cancer screening. And one of those is the fecal immunochemical test. Now, this is a stool-based test. It's a one-sample test that helps detect blood in stool as a proxy for polyps or early-stage colorectal cancer. And this is, in fact, the preferred test for colorectal cancer screening globally and in hard-to-reach populations in the United States. When mailed, the fecal immunochemical test can increase screening participation by approximately 22%. And here I'm showing you what an individual might receive in the mail. And so they might receive an envelope that includes this FIT vial in which that they would use to collect their stool sample. There are some instructions that often accompany these kits. And you can see here that they have the opportunity to write the date on the sample, collect it, and then mail it back. But in the United States, and I should mention that this is a two-step screening process, meaning that if you choose to screen by this test or any other non-invasive test, that if you have an abnormal result, you must go on to complete a colonoscopy. And so here I'm showing you how that test is processed in the lab and usually reads out either a normal or abnormal result. But we know that in the United States, colonoscopy within 1 year of an abnormal FIT rarely exceeds 50% of those who have that result. Our prior work using mixed methods, and here I'm just showing you one of those studies. Here we interviewed providers within a safety net healthcare system, and these safety net healthcare systems provide care to individuals regardless of their ability to pay. And in these settings, individuals often also have higher mortality from colorectal cancer because of lack of access to care. And so here we interviewed providers who took care of these patients and really tried to stratify clinics under the safety net umbrella that were either higher performing or lower performing, and tried to assess barriers that providers were noticing that their patients were experiencing when it came to completing a follow-up colonoscopy after an abnormal FIT. One of the top 2 reasons that we found as barriers to failure to complete a colonoscopy were lack of transportation, which was reported by about 57% of those interviewed, as well as challenges with the bowel prep, which was reported by about 62% of those interviewed. In higher performing settings, when we asked what was facilitating the completion of colonoscopy, we learned that interpretation services was very important, as well as patient care coordinators, which can in many ways serve as physician extenders. As I mentioned, we used a mixed methods approach, and so we looked for similar answers using electronic health records, by doing systematic reviews of the literature, by speaking with patients, and ultimately we found that the barriers to colonoscopy completion from patients, providers, and all these other sources really fell into 3 major buckets. So logistical challenges, personal challenges, as well as financial barriers. And under the logistical barriers, lack of transportation was one that came up often across these multiple methods. So that's the work I'll be focusing in on today. So this is a model that Saeed et al. Presented as a way to relate transportation to access to care and outcomes. And in their systematic review, they reported that in fact, up to 67% of populations face transportation barriers when accessing healthcare. And so one can imagine you have a patient who needs transportation in order to get to timely medical care, including clinician visit, to get timely medication access, including new prescriptions and refills. And here's what I've added to their model that I would postulate transportation is also important for timely procedures and diagnostics, including colonoscopy, but certainly not limited to. And once we are able to help individuals overcome those transportation barriers, you can imagine that we would see improved health outcomes, including improved guideline concordant care, appropriate changes in medication plans, earlier diagnoses, and prevention of chronic disease complications. So we hypothesized that given the ubiquity of rideshare non-emergency medical transportation platforms, such as Lyft and Uber, that these could potentially be scalable and a cost-effective strategy to increase colonoscopy completion. Now, non-emergency medical transportation platforms using rideshare have been used in multiple settings, including primary care, helping patients manage diabetes, as well as in oncology practices, helping individuals get to chemotherapy or radiation therapy. Gastroenterology and colonoscopy are unique because individuals receive sedation, and there is this concern, and often that, if folks receive sedation, then rideshare is no longer a safe option for them. And so we had two aims. First, we wanted to identify the barriers, facilitators, and the process recommendations to implement a rideshare non-emergency medical transportation for patients receiving sedation in a safety net healthcare system. And we started in the safety net healthcare system across our health system because that was where the need was greatest. We then, once we identified these steps, wanted to pilot this program in the population requiring colonoscopy for colon cancer screening. And so because this was an implementation science-focused lecture, wanted to really show you the causal pathway diagram that informed the mechanism of our work. And so we said that our implementation strategy was really the ride sharing, and that we had a couple of implementation strategy options. One could be a large pool of drivers who, would be available to assist these patients get home after receiving sedation. But the other model could be potentially a smaller pool of drivers who would then be trained perhaps in basic life services or in delivering emergency care should anyone experience an adverse event using the intervention. We said that the moderators would patients accept this depending on the time of the day and how easy it was to schedule for patients as well as those in the clinical enterprise. We knew that the preconditions in order to activate this would be one, patients being aware of this program, and then drivers being available. And the mechanism there would be arranging the company transportation really to help overcome the barrier of lack of safe sedation transportation and our proximal outcome, which would really be the colonoscopy completion, but there's even a distal outcome, which would be the percent of eligible individuals who then receive the service. And ultimately that patients would then keep their appointments, they would get sedation and then use this service. So in order to, really implement this, we engaged in informal stakeholder engagement meetings over a year-long period where we had over 22 different data collection point meetings from over 34 individuals. Those included patients, directors of ambulatory care centers, gastroenterologists, nurses, anesthesiologists, as well as the industry partners that we wanted to engage with. We even went all the way up to the state healthcare authority because whenever patients who are under Medicare or Medicaid are involved, there might be some additional hurdles to overcome. We then use storyboards to engage these stakeholders, and then we use the nominal group technique with 5 stakeholders to finalize the workflow. And I'm going to show you next some of these storyboards. Storyboards are a method that can be used to enhance cross-disciplinary communications. And as I showed you, we were communicating with patients, but also with people in industry and then multiple different specialties. And so we would use these in our meetings as a way to demonstrate what the issue is so everyone could quickly arrive at the same page so we could then begin the conversations around using this. And I should also mention that legal and risk management were heavily involved in the conversations based on the initial concerns that I shared. So here we have an example of a storyboard. William, who's a 60-year-old man, who's receiving a phone call from his physician that his FIT test is abnormal and he needs a colonoscopy. Unfortunately, he completes the bowel prep, but in the morning, his wife tells him that she's not able to accompany him because of an unexpected family emergency. So he calls the endoscopy suite and they inform him, and this does happen in real life, that he could either have the procedure without sedation, which is a common entity and some people are comfortable doing so, but also informs him about this potential rideshare program, which he accepts. He arrives at the facility on his own accord. And then once he is done with his procedure, the nurse lets him know that a rideshare has been ordered, that the process is in fact HIPAA compliant, and will be taking him home. So he meets his rideshare driver in the front of the endoscopy unit, and then the next day receives a call from a nurse to check in on how he's doing. Overall, he's feeling well, and he's appreciative of that additional service. So we use these, to have our meetings. And in doing so, we were able to finalize a contract between our healthcare system, the legal risk management teams, as well as our industry partner using this rideshare program. And this is really the study workflow that we all agreed upon after consensus. Was that first, there was already a process in place in which the endoscopy unit would call patients during pre-procedure phone calls to identify patients who didn't have a ride or somebody to accompany them home. Those individuals were then referred to our study team, who would then contact patients to explain the study and obtain informed consent. On the day of the procedure, patients would go through the normal process of the procedure, but if they had provided consent after they had recovered, the nurse would use this HIPAA-compliant platform to order a ride for patients. The patients did not need to have a smartphone. The nurse would often accompany the patients to the front of the building and then assist them in getting into the vehicle. And then once they are in, our study team, as well as the nursing team, is able to track these individuals until they arrive home. And 1 to 2 weeks later, we call them to do interviews to assess their experience. Our primary outcome was really safety, again, because of the initial concerns related to a program such as this. So we assessed, 159 individuals for, there we go. We assessed 159 individuals for eligibility, of whom 41 were ultimately excluded because either we were unable to reach them or they found alternative transportation. Of those, 118 were allocated to the intervention. 15 ultimately did not end up using the rideshare because they afterwards found alternative transportation, either canceled or no-showed to their appointment or were admitted post-procedure. So ultimately 103 people completed the intervention and were analyzed. This is approximately a bit over 60% of those who were initially eligible. So to date, between February 2022 and February 24, 118 patients have been consented. Again, 103 have been completed. 69% are men, 87% speak English, 51% identify as white, 21% as Black, 13% as Asian, 3% as American Indian and Alaska Native, and 15% self-identify as Hispanic. And you can see here that about 45% or 45 individuals are insured under Medicare, about 38 are commercially insured, around 27 are Medicaid insured, and we have about 10 uninsured patients who have participated in this program. Most individuals noted that they did not have any challenges getting to their visits, getting to the healthcare center. Over 80% acknowledged that in an initial survey, but really the issue was getting home after sedation. You can see that the majority lived within less than 30 minutes of the healthcare facility. And when we look at overall usage, the average ride cost was about $36. The median ride distance was 3.6 miles, and the median ride length was about 16 minutes. 100% of patients have gotten to their destination safely and said that they would use the service again and would recommend it to others. And the key barriers that the program has addressed include access to the procedure, where patients noted that they would have had to reschedule or cancel without this option, and autonomy, so not having to rely on an escort, which allowed them for attending to their health needs. And here are some representative quotes from patients. I would want to highlight the second one, where the patient said that there are people like me who kind of fall between the cracks. We don't have any kind of support system, and just having the rideshare made a difference. So here we asked staff members about their perception of the acceptability, the appropriateness, and feasibility of this intervention. And you can see here that overall the scores were quite high, ranging from about 4.5, with the lowest being about 4.3, related to the feasibility of implementation. But overall, the staff that had to implement this were really quite supportive. Someone said, that it was flexible. The patient got dressed, they'd go online, put it in, and the driver would be downstairs within just a few minutes. And they were very impressed with how quickly the service was once initiated. So challenges to consider for broader implementation include professional society recommendations. There are currently lack of consensus on escort guidelines when patients receive sedation, and different opinions when you look at anesthesia societies versus gastroenterology societies. There are certainly legal considerations, but so far not a lot has been published either in formal scientific or legal literature, and it seems to be largely speculative. As I've mentioned, we've now done this in over 100 patients, and everybody has gotten to their destination safely. And then cost. This work has been supported by the NCI and additional programmatic funds. And so certainly thinking about models for programmatic sustainability related to cost are important. So in conclusion, there is a significant and growing need for colonoscopy for colorectal cancer screening and early detection. Transportation is a persistent barrier to colonoscopy completion, including initial screening, follow-up of abnormal non-invasive tests, as well as surveillance of polyps. And addressing transportation barriers can improve overall outcomes and address persistent disparities in colorectal cancer. We demonstrated that in an academic community healthcare system, a rideshare intervention provided by the system was safe and addressed transportation barriers, and that rideshare should really be considered as far as for colonoscopy completion in future CMS coverage decisions. Thank you for the opportunity, and I want to acknowledge my funding sources as well as contact information for those who might have questions later on. Oh, Dr. George, you're muted.
Thank you. I missed my click there, but thank you so much for really your great work. It's really great to see all that you've accomplished, and we will have some questions towards the end after our second presentation, but I really appreciate all you've done. Look forward to our conversation. Great. So our next speaker is Dr. Francisco Tomassoni Ardori, who I'll also ask to pronounce his name properly on his when I'm done. He is a staff scientist and research manager of the gene targeting facility within the mouse cancer genetics program at the NCI. He received his PhD from the Polytechnic University of Marseille in Italy, and completed his postdoc training at the NCI. And as part of the Cancer Moonshot, supports the MyPart program, the My Pediatric and Adult Rare Tumor Network, using his research expertise, developing new mouse models of rare tumors using information gathered from the natural history study of rare tumors. And today he's going to share his work on modeling rare wild-type gastrointestinal stromal tumors, which I can speak from experience is a very uncommon entity and is a really hard disease to model. So I look forward to seeing the progress that you're being made. It's sorely needed.
Thank you very much, Dr. George. And the name is Francesco Tomassoni Ardori. You did very well, because it's tough to say even for an Italian. So let me share my presentation. Okay. I hope you guys can see the slides.
Yes, go ahead, please.
Thank you. So it is really a pleasure for me to be here today with you and share our work that actually was possible thanks to a wonderful collaboration, I would say, between the Mouse Cancer Genetic Program, where I'm working at the NCI Frederick, and the Rare Tumor Network, MyPart, at the main campus NIH. And as Dr. George was saying, we are trying to model a fairly rare gastrointestinal stromal tumor that is known as wild-type GIST. So wild-type GIST are rare tumors, rare malignancies. Technically, they are sarcomas, and they account only for 15% of the total GIST, and they usually occur in the stomach. 60–70% of the GIST occur in the stomach, but they can be found in basically everywhere in the GI tract. And wild-type GIST, unlike the other common GIST, are lacking activating mutation in the c-KIT gene and PDGF receptor alpha. And that's why they are called wild-type, because since these two genes are wild-type, they are denominated wild-type GIST. And this is the reason why these wild-type GIST do not respond, unfortunately, to standard tyrosine kinase inhibitors therapies. And if we look from a genetic point of view, what has been known and what we know so far in terms of mutations that are occurring in these rare tumors, we know that for sure there is a big involvement in what is known to be a mutation in the SDH. SDH stands for succinate dehydrogenase. It's an enzyme that is extremely important, a key enzyme that is present in the mitochondria, in all the cells, and is part of the TCA cycle, Krebs cycle. It is formed by 4 subunits, SDH A, B, C, and D. And these subunits are actually encoded by 4 different genes that are located in different chromosomes. But any mutation that disrupt one of these subunits can actually be deleterious for the function of this enzyme. And other types of mutations that are found in these rare tumors in human are activating mutation of BRAF and loss of NF1 and other very rare sporadic mutations. We can find sometimes like translocation or fusion with FGF receptor or TRAC genes. So our goal was trying to model this rare tumor. And we wanted to test, of course, one of the most common mutation found in these sarcomas, which is the loss of SDH. And to do that, we have generated a conditional knockout mouse model for SDHB by fluxing exon 2 of the gene. And also we wanted to use another common mutation, which is the BRAF V600E activating mutation of BRAF. And our aim was basically to target what they are known to be the GIST cells of they are called interstitial cells of Cajal or Cajal. They are a very peculiar cell type. They are present all around the gut, and they are in the smooth muscle layer that is surrounding all the GI tract. And they are basically mediating the signaling that is coming from the autonomous nervous system. So they are mediators between nervous system and muscle cells, and they're extremely important for gut movements and peristalsis in the gut. So these ICCs, they express 2 important markers. One is c-KIT, and the other is DOG1, that stands for Discovered on GIST1. And these 2 markers are actually used for the diagnosis of GIST. So what we wanted to do was actually target these c-KIT positive ICC, and to do that, we use this c-KIT-CRE inducible mouse model. So this is actually our strategy and how we decided to approach this modeling study. So the first thing we did, we checked if we were able to target efficiently these ICCs. And to do that, we used, of course, our c-KIT-CRE and a reporter mouse model that is known to be the MT-MG. So the way this mouse model works is that upon Cre expression, this tomato cassette is deleted, and basically you can see EGFP signal only in the cells that are expressing Cre. In this case, we are looking for c-KIT positive cells expressing Cre. And we choose this timeline. So we did one tamoxifen injection at P21, so very early in the mouse life, because essentially these wild-type GIST are also known to be happening in young patients in a pediatric setting or in young adults. So we try to stay as close as possible to the human pathology. And so what you are looking here is a stomach slice, and you see the wall of the stomach and the smooth muscle fibers that are surrounding all the guts. Here you can see the stomach here. And in our control mouse, not treated with tamoxifen, we can see that we don't see any GFP signal, but when we use tamoxifen, just a single injection, we are able to spot these Kaggle cells that are perfectly embedded in these smooth muscle fibers. So this was actually telling us that our system was efficient, and we were able to target these Kaggle cells. So next, we basically generated 4 experimental groups: a control group of animals expressing only the Cre, an SDH group where we wanted to conditionally delete SDHB in these Kaggle cells, a BRAF group where we conditionally express the mutant V600E, BRAF V600E. And also we thought about using a combination of the two where simultaneously in the same cells, we are able to delete SDHB and express the mutant BRAF. So we follow this group of animals, about 12 animals per group, for about a year. And while we didn't notice any problem and no tumors developing in the control and in the SDHB group, we actually noticed the first red flag just 3 weeks post-tamoxifen. All the animals that were belonging to these two groups were appearing sick, not moving, hunched, and they had to be sacrificed. And by our surprise, they all showed this phenotype. So basically, the stomach was incredibly dilated and distended. Was a constriction of the pyloric region due to a severe hyperplasia of the smooth muscle layer that is surrounding this area. And actually the pyloric region is per se a sphincter, so it's very rich in smooth muscle cells around. And we can notice also that very often GIST-like lesions were present or figure similar to this, where you can see proliferating cells that are invading the smooth muscle layer. So another step that we wanted to do was characterize these GIST-like lesions that were often present in the pyloric region with these two markers that are known to be expressed in GIST. So we stained for c-KIT and also DOG1, and actually, as you can see, they were both positive in our mouse models. And another important staining was checking for SDHB expression. So when we stain for SDHB, you can clearly see that these lesions are completely well stained and uniformly stained in the BRAF animals, while in the double mutants, you can see that these invading and expanding cells in the muscle layer are completely negative for SDHB. And this was confirming that these double mutant, these cells are actually, negative for SDHB. So as I told you before, in this first study, all of the animals died basically 3 weeks post tamoxifen injection. And these basically spurred us to think about redoing this study and trying to lower down tamoxifen. And the reasoning behind that is that we thought that by lowering down tamoxifen, we could actually target less cagal cells and maybe overcome this critical period of time where the mice appeared all to be sick. But actually, you can see that the SDH loss seems to have an incredible phenotype. Basically, it's worsening the probability of survival of this group. So something that we wanted to do also was to explore in detail the histopathology of all these animals. So what you see here, it's a normal slide of stomach. So you can see zooming in the three most important layers: the mucosa layer, the submucosa, and the smooth muscle layer, the tunica muscularis, where the ICCs are residing, basically. And so we started introducing a grading of histology to grade our lesions, gist-like lesions. And so, as you can see here, in grade 1, you have just focal hyperplasia, so just nodular formation of expanding cells. And going up, grade 2, we consider those like multifocal nodular formation, where you can detect multiple nodular formation along the smooth muscle layer surrounding the stomach. And then grade 3, it becomes always more complex. And as you can see, these expanded cells are fusing together, are coalescing, and there is always less stroma. And as we progress in grade 4, it becomes even worse. So you can see that the presence of stroma is minimal, and these severe hyperplasia is very well expressed here. And then the grade 5 is when we actually detect a gist-like tumor expanding in the tunica muscularis with pleomorphic morphology like seen in human patients. And this is the typical appearance of gist. So we use this grading system to evaluate our studies, basically. And we started evaluating all the animals in the first study that I showed you, where we used 75 mg per kg of tamoxifen. And as you can see, even here, we noticed that in the double mutant, the percentage of mice with gist-like lesion was a little bit higher. And then when we evaluated the other study where we used the lower tamoxifen, we can actually see that these mice in the BRAF group, they don't even have grade 5 lesions, while the other ones are expressing. So overall, there is a tendency to aggravate when there is a loss of SDHB. So basically, SDHB is exacerbating the BRAF-driven phenotype of these kind of tumors. So we also performed another study where we wanted to test the inhibition of BRAF. Clearly, these tumors are BRAF-driven, and we wanted to see, first of all, if dabrafenib, a very common inhibitor for BRAF, was able to slow down these tumors. And also, we did a partial use of this dabrafenib. Instead of treating the animals every day, we decided to go with 3 injections per week in order to slow down but not completely inhibit the BRAF-driven signaling. And thinking about that this strategy would also help us distinguish again between the BRAF and the double mutants. So when we did this study, as you can see, the vehicle-treated animals behaved essentially like I showed you before, and they died more or less 3 weeks post-tamoxifen injection. But when we used the BRAF inhibitor, we could extend the lifespan of these animals. And again, we see that the double mutant, the loss of SDHB, it has an effect on the probability of survival of these animals. Also, we characterize the histology of these animals here. And again, you can see that the vehicle, they show the higher grade of a severe hyperplasia and GIST-like tumors, while the dabrafenib-treated animals show a significant difference between the two groups. And again, we see that the loss of SDHB is promoting higher grades of pathology. So loss of SDHB basically is responsible for many molecular alterations. And the reason why is because there is an accumulation of the substrate of this enzyme, which is succinate, and higher levels of succinate, that is also known to be a non-co-metabolite, is first of all inhibiting proline hydroxylases and causing what is known to be a pseudo-hypoxic state, stabilizing HIF-1 and 2 alpha, and also is inhibiting the TET family of enzyme, and so is causing hypermethylation of the DNA. Again, hypermethylation of DNA is also has been reported in human GIST to be responsible for an abnormal topology of the chromatin. So what essentially is happening is that the normal topology and arrangement of the chromatin, when there is hypermethylation, can be disrupted because of these CTCF unpairing. And so those regions that are known to be insulator of these sort of looping. When CTCF cannot pair, these enhancers can actually activate these genes. And in case of GIST, FGF4 has been reported to be one of these, so an overexpression of an oncogene, if you will. So we decided to go after this molecular alteration and check if our mouse models were expressing these. So we analyzed pyloric regions of these animals, basically 20 days post-tamoxifen, just before the animals are getting sick. And as you can see, when we checked for HIF-2 alpha expression, we could see an upregulation and stabilization of the HIF-2 alpha protein compared to the control mice. We also checked for HIF-1 alpha, but we couldn't see any difference. And also we checked, again, using pyloric region 20 days post-tamoxifen by conducting mass spec analysis. We checked the hydroxymethylcytosine levels that directly indicating the methylation of DNA. So we could detect a significant drop in the 5-HMC levels in these pyloric regions, indicating that these animals actually have a hypermethylation of DNA. And also we confirmed this by histology, by using an antibody able to recognize 5-HMC. And as you can see here, while the BRAF animals, they show more or less a uniform expression of 5-HMC in the nuclei, you can see that in these SDHB, BRAF SDHB double mutant, many nuclei don't show any staining for 5-HMC. So this was confirming that this hypermethylation is actually present and playing a role there. And following this, lastly, we checked for FGF4, which was reported to be upregulated in human GIST. And again, these mice are showing that the pyloric region 20 days post-tamoxifen are expressing a higher level of FGF4. So basically, summarizing, we were able to target these c-KIT SDH-B, basically SDH. We are able to originate tumors that are recapitulating many distinctive features of wild-type GIST. And I would say also that another important observation that we have done is that the SDH-B loss alone is not sufficient to induce GIST-like tumors, at least in our mouse models. But we can clearly see that it seems to exacerbate the BRAF-driven GIST that we have seen here. And basically, we have seen that there is an effect on viability and survival of these animals. We have seen a higher methylation status in these animals, and also higher levels of FGF4 and IF2 alpha. So now what we are doing is we are trying to establish allografts using these mouse models. We are taking the pyloric region and try to transplant these pyloric region in B6 mice to see if we can have a more pure population, because our problem is that we have a very heterogeneous tissue at this point. And also another thing that is brewing in the lab now, and that we are testing, and I'm very excited about this, is we found that a tumor-associated receptor, CD276, is actually expressed in these tumors when these tumors are developing. So we are lucky to have Dr. Sancroix's lab next to our lab. So we are testing the efficacy of these ADC. So we are targeting with the antibody able to recognize CD276 with an antibody drug conjugate approach. And this experiment is actually ongoing as we speak. So we should know about this very soon. And with this, I want to thank all the people that are involved in the in our lab. Colleen has been doing a terrific job in maintaining the colonies, all the mouse cancer genetic program, people that have been involved in the generation of our SDH-B mouse model, Dr. Sancroix's lab for the CD276 study, all the MHL for the great support, and our collaborator, Sauer from the Technical University of Munich, that provided us the c-KIT inducible mouse, c-KIT inducible mouse model, and the CAPER. We are collaborating with them for generating allograft, and of course, my part. It's a huge group, but I just want to thank Brigitta, Carleen, and John that are the most involved person in this project. And thank you all for the attention, and I will be happy to take any questions or suggestions you may have. Thanks.
Wonderful. Thank you so much for that wonderful talk. I think we have about 10 minutes left for discussion and questions. You know, we're really lucky because you both presented such wonderful talks. It's such different ends of the spectrum of how we think about progress that needs to be made in cancer care, in cancer biology, all the way to cancer prevention, really, or early detection. So it's really a wonderful expression of the support of the Moonshot programs across this continuum. So I actually have been taking a look at the chat. Thanks to all of you who have entered questions. Feel free to continue to do so. I'm going to start by asking Dr. Isaka. One of the themes in some of the questions is around scalability in rural environments, and curious about your thoughts there of your program.
Yeah, I think a program like this can actually be scaled in rural settings. I would say Seattle is a very expensive market. It's one of the most expensive places to live in the country. And if you look at the cost of our rideshare, you know, I mentioned it's still on average $36 per person. Whenever I travel, I'm shocked at how affordable rideshare is. So I imagine that in rural settings, yes, there might be fewer availability of drivers, but it's actually going to cost less, which will allow you to reach more individuals.
And can I ask a question about the study design? I may not have appreciated it. I believe that the focus of your results was having patients get home after. How did they get there?
That's correct. So as I mentioned, over 70% of people said they did not have a difficult time getting to the healthcare system. So most would either walk, take the bus, other public transportation methods to get to the facility. And that's how they engaged in most of their care. It was really the challenge after sedation, getting home. That was the issue.
Great. And then I'm going to ask you another question before I head over to our mouse model side. One question that came up also were implications for other screening modalities. And or you talked a little bit in your background about treatment, but how do you think that this may affect other screening modalities?
Well, you know, for colon cancer specifically, I'm a co-author on a paper that was recently published on a blood-based test for colorectal cancer screening. And as I mentioned, with all non-invasive screening strategies, there are 2 steps, meaning anyone with abnormal results will need a colonoscopy. And so if we anticipate that blood-based tests such as colorectal cancer screening, or even the multi-cancer early detection assays, you know, as we know, coming down the pipeline, if you have individuals with abnormal results, they're going to need follow-up. And when it comes to colonoscopy specifically, they're going to need sedation. That might be true also for the abnormal lung cancer screenings. And so I think a program such as this really addresses those transportation barriers for the sedation piece that's needed to follow up screening tests. And as I mentioned, this rideshare has otherwise been used in primary care settings and even oncology settings already where sedation is not administered. So there's really quite a potential to scale this across multiple screening types.
Wonderful. No, that's great. Okay, so Dr. Tomasini Ardori, several questions. Thank you again, both for your excellent presentations. But there's a question saying SDHB loss is insufficient to drive tumors in your models, but it does appear to be sufficient in humans. I wonder your thoughts about that question, and any comments that you have.
Yes, that's actually one of the things we are discussing a lot in the lab, because so far what is known in human is that SDHB seems to be the only one present in these tumors. So the thing is that probably either in mice, SDHB loss is not sufficient to drive tumors, or what we think actually is that maybe there is something else together with SDHB, even in humans, that we are missing. And this is something we are trying to address, honestly. And so in our models, we looked at the mice up to 1 year, and when you look at the histology, basically there's no difference between the control and the SDHB loss. Another theory could be that since it is a very important gene, when you delete SDHB, you may end up killing the cell. So maybe there is the need of something else pushing the survival of the cell together with the loss of the SDHB, and is a sort of adjuvant gene that pushes the oncogenesis process. We still don't know this, but we are actually very interested in looking into this.
I think it's interesting to make a comment that many patients and people that have SDH deficient GIST have germline mutations in SDH, in one of the SDH subunits. Yes. And that it appears to be that second hit of something, of either the second loss or something that leads to these SDH associated neoplasms, and whether it's GIST or paraganglioma or a different SDH altered tumor. So it is interesting that there does appear to be this germline component that contributes, that suggests that some second issue needs to occur.
And actually, I would add also that, as you said, SDH loss is also present in other rare tumors like pheochromocytoma, for example. And there are a couple of studies where they knocked out SDH to study these tumors using different CRE drivers. And they couldn't notice as well any development of tumors until they use something else. So there is a recent study where they use a loss of NF1 and loss of SDH, and they are able to mimic pheochromocytomas, but SDH alone is not sufficient. So it's a sort of another evidence that SDH alone is not sufficient. So maybe even in humans, we should be looking a little bit better, because when you look after, since SDHB is causing so much DNA hypermethylation, there could be so many events that we are seeing that are, not the origin of the tumor.
Great. And Dr. Isaka, Leah Mechanic from the NCI raised an early question, noting that one of the barriers for colonoscopy was the prep, and wondered if you had any thoughts about interventions to address the barrier of a colonoscopy prep.
Yeah, so a lot of the barriers that we've identified and others have identified has to do with the volume of the prep and the instructions related to the bowel prep itself. So there are lower volume bowel preps that are currently available on the market. They just may cost individuals more out of pocket. So it's sort of addressing those financial barriers related to that. And then for those for whom it's more of the instructions, it's really trying to streamline the instructions around the bowel prep to make it as simple as possible for people who need to complete it prior to doing the procedure.
Excellent. And then a clarification on your methods. Kelly Fletcher asks, did you use the large or small pool of drivers in your pilot program?
Yes. So we approached the rideshare company and we learned that unfortunately you cannot modify the existing product in a market. And so for our Seattle market, we did not have access to a smaller pool of drivers. We ended up using the larger pool of drivers. And as I mentioned, the modification was that a staff member could then accompany individuals to the front of the building for patients to assist them to get into the vehicle, and then we track them from there on.
Wonderful. And I have a question actually about the insurance. I know that you mentioned bringing in people that were on Medicare and Medicaid, and then bringing in your state authorities. What was that like? What kind of feedback did you get? Tell us about that a little.
Sure. So you'll see that when I showed the number of insurance people was lower for the Medicaid insured population. And that was because initially our state healthcare authority told us we could not offer this to Medicaid insured individuals. They were concerned about double dipping. And we in fact sort of expressed that this is just an option because the existing programs for those groups were very unreliable. Often, when canceled, led to really long waits and overall high dissatisfaction. And so we went back after an initial conversation, and when we did, they said, okay, you could then offer it to them. And we saw a huge bump in people who were Medicaid insured then using the service. And so it's available for all. And, that's kind of why I mentioned the need for CMS coverage rules that include this. And we have work that we're going to be presenting in an upcoming conference that's a cost effectiveness analysis that answers this exact question and trying to move that policy forward.
Wonderful. Thank you. Okay, Lillian.
Oh, thanks so much to Drs. George, Isaka, and Tomasino Ardori for a wonderful webinar. We have more questions that we didn't have time to get to, but we will be sharing those with the speakers. And I guess I will close by thanking our audience for your participation and highlighting our next Cancer Moonshot seminar, which will be on May 23rd. This will be a unique seminar in that it's more of a Cancer Moonshot conversation on the topic of advancing data sharing through the Cancer Moonshot. And we'll have discussants from Drs. Kai Tan and Liz Salmi, who will be sharing their perspectives on data sharing, both as a data user, generator, and the patient perspectives. Thank you all and have a good day.
U.S. Department of Health and Human Services, National Institutes of Health, National Cancer Institute, cancer.gov, 1-800-4CANCER.